The idea is to talk about nucleoside analogues, which are used, for example, to treat viral infections of the type like herpes, HIV, hepatitis. So, how nucleoside analogues work? As the name may be hint for, nucleosides in the body become nucleotides. Nucleotides are the building blocks of DNA and RNA. The difference is uh, T and U in DNA in comparison to RNA. Anyway, they are the building blocks of DNA and RNA. As you know, T, G, U, and all of this. The double strand is built from nucleotides. But nucleoside analogues, what they do two main things. They incorporate it in DNA and RNA, and they cause chain termination. This is the one mechanism of action. And the second thing is inhibition of enzymes. So chain termination, how they do that? They incorporate into DNA or RNA as nucleoside analogues, and they don't let the enzymes elongate the DNA or RNA. So the DNA or RNA are stopped from elongating and replicating, so the cell cannot create RNA and cannot create proteins. It cannot use the DNA, so it dies. Uh, and the second mechanism, inhibition of enzymes, especially enzymes that are important for DNA functioning. For DNA, as you can see here, nucleotide acid synthesis. So again, it damages the DNA. There is also another thing that those drugs, nucleoside analogues, are selective to the viral cells and not to normal cells of the human, therefore reducing their toxicity to normal cells. And you can see here that acyclovir works quite the same way. Acyclovir is used for viral infections like varicella zoster virus and herpes simplex virus. You can see here the selective activation in cells of the herpes, convection to the active form, and then inhibition of viral DNA polymerase. It incorporated into the viral DNA, and it caused termination of the elongation of the DNA, and it goes selectively to a viral enzymes. Okay, so another thing is how they inhibit those enzymes. They can cause direct inhibition, but let's look at the picture. This will be maybe more understandable. So you can see the nucleoside analogues come and enter the cell, and then they get the two phosphate groups, and then they can incorporate into the DNA or inhibit enzymes, and therefore inhibition of viral replication. This is the main mechanism of nucleoside analogues. Okay, and that's it. Thank you very much. If there are any corrections, please correct me. Thanks Copilot, thanks for the images, and that's it.